Hello friends, good afternoon and welcome to EduSite live lectures. Dear friends, today in immunology we will be talking about cells of immune systems. To discuss this topic we have with us our subject expert Dr. Hardeep Kaur. Dr. Kaur is assistant professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you, Amrit. Hello viewers, once again we are here to discuss about immunology and a very important topic in immunology, the cells of the immune system. Now viewers, I am sure that you must have understood how important an immune system is in our body. Any kind of pathogen, be it a bacteria, virus, fungi, parasite, anything with which we are inflicted with, we are protected, we do not know about what is affecting us because of a very efficient immune system within us. We also realized that a hyperactivity of an immune system or an underactivity of an immune system, both are very deleterious to us. We have to maintain an effective balance. It is the function of the immune system to have a very important balance within our body to ensure that all kinds of pathogens are devoured or are killed within our body. Now, coming to the main important force of the immune system, the cells of the immune system which are responsible for providing an effective protection within us, let us come to this topic. Now, after this lecture, you will be able to understand what is hematopoiesis, the hematopoietic stem cells, how hematopoiesis is different from apoptosis, and finally, the cells of the immune system. Now, coming first to hematopoiesis, what exactly is hematopoiesis? The production of all types of blood cells including formation, development and differentiation of blood cells. This is the definition of hematopoiesis from Medicine-Net, which says that all blood cells, whether it is a RBC, WBC, platelets, how they are formed, they undergo development and further differentiation, they all are coming under the process of hematopoiesis. Now, we must understand that prenatally, like for example before the child is born, when it is in a fetal state, the hematopoiesis takes place initially in the yolk sac, then in the liver and spleen and finally in the bone marrow. This is what takes place within the fetus. Once the fetus is born, almost all hematopoiesis takes place in the bone marrow. In normal situation, hematopoiesis in adults occur in bone marrow and lymphatic tissues. All types of blood cells are derived from primitive cells which we call them as stem cells that are pluripotent. Now, what exactly we mean by pluripotent? That they have the potential to develop into all types of blood cells. Now, pluripotent is different from unipotent. When I talk about a unipotent cell, unipotent means the cell has a destiny. It will go into and form a particular type of cell only. While on the reverse, pluripotent cell is a type of cell which can develop into any type of cell. So, a unipotent cell has one destiny while pluripotent cell can have many destiny. It can become any type of cell. So, when we are talking about hematopoiesis and when we per se talk about hematopoietic stem cells, they are those cells that are pluripotent and can form erythrocytes also, they can form platelets also, they can form leukocytes also, any type of cells, any type of blood cells they are able to form, hence called pluripotent stem cells. Now, the site of hematopoiesis, as I just discussed, in fetus, the entire hematopoiesis can be divided into three major stages. One is called the mesoblastic stage, which lasts from 0 to 2 months of the embryonic life. Here what happens? Here the cells are formed, the blood cells are formed in the mesenchyme of the yolk sac, which is outside the embryo. So, the entire process of hematopoiesis is taking place outside the embryo. The next stage is called the hepatic stage, which lasts from 2nd month to 7th month onwards. Here hematopoiesis exclusively takes place in liver and finally into spleen. Initially it starts at the end of the 2nd month, maybe like 7th to 8th week. The hematopoiesis begins in the liver, eventually it goes into the spleen. So, from 2nd to 7th month, the hematopoiesis has shifted from yolk sac outside the embryo to the liver and spleen of the embryo. And in the final stage, which is called the medullary stage, this takes place from 5th month of gestation onwards, when the bone marrow is exclusively involved in hematopoiesis. Now, this takes place during the fetal development or during the gestational period. Now, if we see the slide, we will see in infants the hematopoiesis exclusively is taking place in bone marrow and on bones, while in adults we have hematopoiesis in the vertebrae, ribs, sternum, sacrum, pelvis, femur, the proximal end of the femur. These are the places where continuous hematopoiesis is taking place in adults. Now, as you can see in the graph, on the x-axis is the months, gestational months, and on the y-axis is the rate of hematopoiesis. As you can see, from the beginning to the end of, let us say, second or the beginning of the 3rd month maybe, the exclusive hematopoiesis is occurring in yolk sac, which is outside the embryo. This yolk sac is present is an extra embryonic membrane which is present outside the embryo. The entire production of blood cells is taking place in the yolk sac. While as you can see, after once the 2nd month gestation has the landmark has reached, the hematopoiesis starts in the liver, in the fetal liver. This is followed by hematopoiesis, and as you go further after the 3rd month, you will see the hematopoiesis is taking place in spleen also. So, this is the second phase or the hepatic phase where hematopoiesis is taking place in the liver and the spleen of the fetus. As you go further after 5th month, as you see there is a plateau is reached. The maximum hematopoiesis has taken place in liver and spleen. Now, the onus of hematopoiesis has gone on to the bone marrow in the fetus. Now, 5th month onwards, the hematopoiesis level increases in the bone marrow till the birth. And once the fetus, or maybe the we can call it as a neonate, the entire hematopoiesis in neonate, that is the newly born child, is taking place in bone marrow and will take place in bone marrow for his entire life. Now, what exactly is hematopoiesis? Hematopoiesis, as I just told you, it is the production or formation of blood cells, formation, differentiation, etc. Now, it is an entire process which involves hematopoietic stem cells. As I told you, they are pluripotent cells. The hematopoietic stem cells actually they differentiate into the pluripotent cells, these are the multipotent or pluripotent cells which finally get changes into progenitor cells. Now, once these stem cells or the hematopoietic stem cell HSC as we call them, they change into progenitor cells, they lose their potency or pluripotency. So, let us define what exactly hematopoietic stem cells are. HSCs, as you can see in the slide, is a multipotent or pluripotent cell which is able to differentiate into erythrocytes, granulocytes, monocytes, mast cells, lymphocytes, megakaryocytes, any kind of blood cell. Now, they are few in number. Now, for every 5 into 10 raised to the power of 4 cells, blood cells or the cells present in our blood, in our vascular system, there is only one HSC present. That means, for every 5 into 10 raised to the power of 4 cells, blood cells, we have one HSC. Their number are very few. Now, that becomes very detrimental for us to understand these HSCs in vitro condition. One, it is very difficult to grow HSC in vitro. Second, their number is so small. Now, is the number always so small? No. Generally, the number is maintained at a stable level throughout our life. But whenever there is a need, so it is a need based thing. Whenever there is a need, the HSCs start proliferating and develop into a particular cell line. So, we can say they are fewer in number, they are difficult to grow in vitro, however, they are maintained at stable levels throughout our life. Now, events during hematopoiesis. Now, what exactly happens during hematopoiesis? A multipotent stem cell differentiate into one of the two pathways or lineages. One is called the common lymphoid progenitor cell or the common myeloid progenitor cell. Technically, we have an HSC. The HSC has a fate, it can either get into the myeloid lineage or it can get differentiated into a lymphoid lineage. Now, this progenitor cell, as I told you before, they have lost the pluripotency capability. Now, the common lymphoid progenitor cells which are formed, the lymphoid lineage, would always exclusively give rise to B lymphocytes, T lymphocytes or the natural killer cells and certain dendritic cells. However, the myeloid stem cells will generate progenitor of RBCs, that is erythrocytes, and many of the other white blood cells, for example, the neutrophils, eosinophils, basophils, technically the granulocytes and the monocytes, mast cells, dendritic cells, platelets, etc. So, we have two lineages. One, as you can see in this slide, it is very clearly depicted. We have a multipotential or multipotent hematopoietic stem cell heading the flow chart. These are hemocytoblast, also called hemocytoblast. As you can see in the arrow, this arrow means that they are self-renewing. They grow on their own, they grow into clones of their own. Now, they have two lineages. They can either become common myeloid progenitor cell or they can develop into common lymphoid progenitor cell. As I just told you, the common lymphoid progenitor cell has lost its pluripotency. Now, it can only form lymphocyte. They can form small lymphocytes or the natural killer cells. The small lymphocytes can further differentiate into B lymphocyte or T lymphocytes. The B lymphocytes can actually become plasma cell or the memory cell. Now, on the other hand, if you see, the common myeloid progenitor cell can differentiate into an erythrocyte, that is the RBC. It can develop into a megakaryocyte. Megakaryocyte is a precursor of thrombocytes or blood platelets, or it can become a mast cell, or it can differentiate into a myeloblast. Now, myeloblasts are mostly forming granulocytes or monocytes. Now, granulocytes can be eosinophils, basophils, neutrophils, and of course, the myeloblast can also form the monocyte. The monocyte can itself differentiate into macrophage once it goes into the tissues. So, this is the entire scheme of hematopoiesis, where the hematopoietic stem cell which is pluripotent is converting into the progenitor cell, myeloid or lymphoid, and the lymphoid leading to the lymphocyte production, while the myeloid lineage leading to all other cells of the blood. Now, if you see in this slide, we have shown that these particular cells, for example, the neutrophils, the eosinophils, basophils, they are mostly circulating in the blood. Similarly, the monocytes, T lymphocyte, B lymphocyte, natural killer cells, platelets, RBCs. However, if we see their counterparts, the eosinophils not only can be circulating in the blood, but they can percolate into the tissue to form the tissue eosinophil as well. Similarly, the mast cells are exclusively found in the tissues. They are always found in the tissues. The macrophages, if the phagocyte is present in the circulating blood, we generally call it as a monocyte. However, when it infiltrates the tissue, we call it as a macrophage. The T lymphocytes are not only present in the circulation, but are also present in the tissue. The B lymphocytes generally are present as plasma cells. Plasma cells are the factories producing lots of antibodies. The natural killer cells, again, they can also be present in the circulating blood and also present in the tissue. Now, what defines that a hematopoietic stem cell should take a lymphoid lineage or it should take up a myeloid lineage. What defines that? It is said that these HSCs, the hematopoietic stem cells, are highly influenced by HIM. That is, HIM is a short form or an abbreviated form for hematopoietic inducing microenvironment. Now, where exactly this HIM is present? Now, these hematopoietic stem cells are generally which are present in bone marrow, because as we said, the hematopoiesis in adults is taking place in bone marrow. Now, hematopoietic cells, when they are present in bone marrow, they undergo further growth and maturation on a meshwork of non-hematopoietic cells, which we call them as stromal cells. Now, what are these stromal cells? These are, as I just said, non-hematopoietic. They are not directly involved in making blood cells, but they are influencing the making of the blood cells. They could be fat cells, they can be endothelial cells, they can be fibroblasts, they can be macrophages. All these cells are forming a mesh within the bone marrow, and hematopoietic stem cells, that is the HSCs, are engulfed or are present within the meshwork formed by these stromal cells. Now, the beauty of these is these stromal cells, they not only support the growth and differentiation of HSCs, but also they influence the differentiation of HSCs also, as they produce certain hematopoietic inducing microenvironment which consists of a matrix, a cellular matrix, and certain growth factors which are important for differentiation of these HSCs. Now, what are these HSCs? As I just told you, one HSC can give rise to 10 raised to the power of 6 mature blood cells after 20 divisions. Whenever there is need, HSC respond to the growth factors. Now, what are these growth factors? These are the growth factors which are secreted in the HIM by the non-hematopoietic stromal cells. Now, HSCs influence the growth factors produced by these stromal cells, influence the HSC to take up a particular lineage and start dividing accordingly. Now, they can lead to increased production of, whenever there is a requirement, it can lead to production of a particular cell line. For example, we need a particular lymphocyte, a B lymphocyte at a certain point in our life. Now, the growth factors in the environment of HSC will influence the HSC to produce the B lymphocytes accordingly. Now, what exactly are hematopoietic growth factors? The hematopoietic growth factors are those factors which are produced by hematopoietic inducing microenvironment. Now, these growth factors are like IL-1, IL-3, IL-6, GM-CSF and many other factors. Now, what do these factors do? These factors influence the production or the lineage taken up by HSC. For example, if a granulocyte colony stimulating factor is produced in the microenvironment, it will influence the HSC to develop into that particular lineage of granulocyte. Or if IL-3 is produced by the microenvironment, it will influence the HSC to take a particular lineage. For example, as you can see in this slide, the multipotent hematopoietic stem cells, if they are exposed to HL1, HL3 or HL6 and also GM-CSF etc., they will influence it to go into the myeloid lineage. That means the HSC will convert itself into a common myeloid progenitor cell. Further, now if it is given GM-CSF, it will become a myeloblast, and myeloblast, as I told you, will either become a granulocyte or a monocyte. That is, it can become basophil or a neutrophil or eosinophil or a monocyte depending upon what growth factor it is being given. Similarly, if it is a common lymphoid progenitor cell, if it is given a TNF alpha, IL-2, interleukin 2, interleukin 7 or tumor necrosis factor alpha, it will form a small lymphocyte. Now, again, the small lymphocyte can differentiate into P lymphocyte, or if it is exposed to another cytokine like IL-1, IL-2, IL-4, IL-6 etc., it will develop into T lymphocyte. So, I hope you have understood that growth factors which are present in the HIM, that is hematopoietic inducing microenvironment, will finally have an influence on the HSC to take up a particular lineage and form a particular cell. It is all a very regulated mechanism. These growth factors are influencing this HSC to finally become either a lymphocyte or any other blood cell. Now, further, it has also been found that not only these growth factor, but the genes present in the cell are also regulating the hematopoiesis. Now, most of this has been done by the disruption studies, where gene knockout technique has been used. A particular gene has been knocked out and seen whether what influence the knockout is having, what it is leading to, which cell is not now produced. A gene, if it is knocked out, a particular cell is not produced. So, we can say, decipher that this gene is probably regulating the production of this particular blood cell. So, certain transcription factors have also been found to play a key role in the process. These are GATA1, GATA2, PU1, BM11, Icaros, OC2. Now, as you can see, few of the factors are very have a specific lineage. For example, GATA1 will only lead to erythroid lineage, while GATA2 can also influence erythroid, myeloid and lymphoid lineage also. So, technically we have various kinds of transcription factors. Some are very specific, some are not as specific, but these transcription factors have been found to have a regulatory effect on the process of hematopoiesis. Now, what exactly, how can I say that hematopoiesis is regulated in naturally also? We have a cell production in our body. The cell production is always equivalent to the cell destruction. For example, the average lifespan of a RBC is 120 days. Now, the various white blood cells have lifespan ranging from few days, for example, in neutrophils, to as long as 20 to 30 years for some T lymphocytes. Now, for maintaining a steady state level, the average human being must produce an estimated 3.7 into 10 raised to the power of 11 WBCs per day. Now, if we are producing this many cells every day, if all these cells live forever, then it will be a very dangerous situation. It is almost like a transformed state in our body. So, the cell production is always going hand in hand with cell destruction. A programmed cell death, which we call apoptosis or apoptosis sometimes, it is a very regulated process which involves the activation of intracellular proteins in the cell shrinkage, condensation of chromatin material, nuclear fragmentation, cleavage of intranucleosomal genetic material, and finally the cell death. The naturally occurring programmed cell death or apoptosis ensures that we have a regulated number of cells in our body. So, technically hematopoiesis is going hand in hand with apoptosis, which regulates the number of blood cells. Now, in a normal adult, the blood cell count, as you can see, the red blood cell count is almost 5 into 10 to the power of 6 cells per mm cube. The platelets are 2.5 into 10 to the power of 5 cells per mm cube. The leukocytes of the WBCs are enormous. They are 7.3 into 10 to the power of 3. Now, when we are talking about leukocytes, we have to understand that leukocytes are of various kinds. They are neutrophils, they are lymphocytes, they are monocytes, they are eosinophils, they are basophils. If you go back to this table, you will see the percentage of WBC varies. We have maximum number of neutrophils in our body. In fact, they form the first line of defense in our body. Neutrophils are about 50 to 70% in our body, followed by lymphocytes, which are 20 to 40% in our body. This is followed by monocytes, which are 1 to 6% in our body, eosinophils 1 to 3% in our body, while basophils are less than 1% in our body. Now, if you collect the blood from a mammal or a human being and centrifuge it at a high speed, you will find that there would be a non-cellular component which is called the plasma, and the cells will start aggregating downwards in the test tube. The 45% of the blood is composed of RBCs, while the plasma is composed of the 55%. In between you have a buffy coat which is composed exclusively of white blood cells and platelets. Now this is the situation after centrifugation of the blood. The above plasma does not have any blood cells. Now further going into the details of the cells of the immune system, we will find that the cells are of two types. Certain cells which are having a big role in the innate immune system, while the other cells which have a role in the adaptive immunity. The cells which have a tremendous role to play in the innate immune system are the granulocytes, which include eosinophils, basophils, neutrophils, natural killer cells also, certain macrophages, the mast cells, dendritic cells. While on the other hand, the cells involved in the adaptive immune response are exclusively lymphocytes, including B cells, T cells, natural killer cells, etc. So these together ensure that we are protected in our body. They all form a basis of the immune system and provide an exclusive protection to our body, either through the innate system or through the adaptive immune system. We will go into the details of the cells of the immune system in our next lecture. Thank you very much. Welcome back viewers. In this particular session, we will be dealing about the cells of the immune system. In our previous lecture, we had defined what hematopoiesis is, what are the hematopoietic inducing environment, and what are the key role played by apoptosis or apoptosis to regulate the hematopoiesis. Now coming exclusively to the cells of the immune system, as you can see in this slide, in a normal adult blood cell count is as follows. The RBCs are 5 into 10 to the power of 6 cells per mm cube. The platelets are 2.5 into 10 to the power of 5 cells per mm cube, while leukocytes or the WBCs are 7.3 into 10 to the power of 3 cells per mm cube. Now when we are talking about WBCs or leukocytes, as I told you previously, they are of 5 types: neutrophils, lymphocytes, monocytes, eosinophils and basophils. And when we define percentage wise, the neutrophils occupy 50 to 70% of the blood cells, while the lymphocytes are forming 20 to 40% of the WBCs present in the blood. Monocytes occupy 1 to 6%, while eosinophils are 1 to 3% and basophils are less than 1% of the WBCs present in the blood. We had explained this in the previous lecture, wherein we said that if the blood is centrifuged, we will find the blood is made up of a non-cellular component called plasma. The RBCs are predominant, while the buffy coat which is present after the centrifugation is composed exclusively of white blood cells and platelets. We had also explained that out of all the blood cells, few cells are involved in the innate response while few cells are involved in the adaptive response. For examples, the granulocytes, for example, basophils, eosinophils, neutrophils, and the mast cells, dendritic cells, macrophages, that is the phagocytes, are exclusively involved in the innate immune system. That is, they form the first line of defense of our body. I hope you remember the innate immunity is something which we possess by birth, by virtue of our genes, we are the genetic makeup we are made up of. This innate system is present by birth. So, these cells are the important cells of the innate immune system. While the B cells, that is B lymphocyte, T lymphocytes and natural killer T cells are present in the adaptive immunity. This adaptive immunity is a slow response which comes once the innate immunity has already taken place in the body. Adaptive system, our immune response is, our immune system is involved in it. In innate system, it is defined by the genes we are composed of. So, now coming back to the cells of the immune system, the innate system, that is what we are born with. The main cells of the immune system which forms the innate system are the phagocytes. The PMNs, that is polymorphoneutrophils, neutrophils, monocytes, macrophages, the natural killer cells, basophils and the mast cells, eosinophils and the platelets. They are the very important cells of our innate system. Now, you will understand that out of this platelets and RBCs, if we keep it separately, the rest all are WBCs. WBCs, as we all know, we had read this numerous times that WBCs are granulocytes, agranulocytes. Granulocytes are eosinophils, basophils, neutrophils, while agranulocytes are monocytes, macrophages and lymphocytes. Now, out of these, you must understand that lymphocytes are the ones which are responsible for the adaptive system, while rest all the lymphocytes are somewhere down the line are having an ancillary role, they are having an accessory role. Lymphocytes are the main cells of our immune system. The other cells, whether it is the monocytes or the macrophages or the eosinophils, neutrophils, basophils, they all have some other role to play. They are helping, engulfing bacteria, doing something in the body, secreting something, but the main function is done by the lymphocytes, and lymphocytes belongs to the adaptive immune system. The rest all belong to the innate immune system or the first line of defense of our body. Now, coming to individual cells, what is the phagocyte? As I told you, phagocytes are cells that protect our body by ingesting or phagocytosing the harmful pathogen. The harmful pathogen could be a foreign particle, a bacteria, or it could be a dead or a decaying cell also. The name phagocyte comes from the Greek word phagein, which means to eat or to devour, cyte, which means the cell, and the Greek word kutos, meaning hollow vessel. Now, they involve the PMNs and the neutrophils as well as the monocytes and the macrophages. Now, coming first to the PMNs and neutrophils, as you can see, PMNs, neutrophils, they constitute about 50 to 70% of the circulating WBCs. Neutrophils are produced in the bone marrow from the myeloblast type stem cell, which is called polymorphonuclear cells. Now, why is it called polymorphonuclear? As you can see in the slide, the nucleus is multi-lobed in this. Their main role is in inflammation. They leave the blood or the endothelial layer and go into the tissue. The process is called extravasation. The neutrophils are attracted in the tissue by certain chemotactic factors which get stimulated only on tissue damage. So, whenever there is a some tissue damage, these chemotactic factors will be released. These chemotactic factors will influence or attract the neutrophils to the place of injury, And once these factors could be the complement proteins, these could be the clotting proteins, these could be T cell derived cytokines, all these chemotactic factors which are released at the place of injury will influence or will attract the neutrophils to reach at the site of injury. Once it reaches the site of injury, it phagocytoses the pathogen which has come there. So, phagocytic process which the neutrophil is involved, it destroys the ingested microorganism via either through the oxygen dependent mechanism or oxygen independent pathways. So, we can rightly say that they are the first line of defense, they are the first to arrive at the infection site. Now, after this, the important cells which are phagocytic cells are the monocytes and the macrophages. They are also called the cells of the RES system or the reticuloendothelial system. They include the circulating monocytes, these resident macrophages in liver, spleen, lymph nodes, thymus, some mucosal tissue of the respiratory and alimentary tracts, bone marrow, connective tissue, etc. Now, you must understand, we had discussed it previously also in our one of the previous slides, that monocytes are always circulating in the blood. If it is blood borne, it has to be a monocyte. If it is blood borne, it is phagocytic, it has to be a monocyte. If it is tissue borne, we call it as a macrophage. Now, based on which macrophage we are talking about, whether we are talking about a lung macrophage, we name it as an alveolar macrophage. If we are talking about, let us say, a brain macrophage present in the brain, we call it as a microglial macrophage. If it is present in the peritoneal cavity, we call it as a serosal. If it is present in the liver, we call it as a Kupffer cell. So, depending upon its location, its nomenclature has developed. So, various macrophages in various tissues have been given different name. So, we must understand monocytes are blood-borne phagocytes, while macrophages are tissue-borne phagocytes. Their function is same, that is phagocytosis of the pathogen. Now, there are certain other macrophage-like cells also present, which include dendritic cells like cells present in the lymph nodes, present in the, let us say, the Langerhans cells in the skin, the glial cells in the central nervous system. They also have a similar function to the macrophages, that is, they are also phagocytic in nature. They are not macrophages as such, but they are very similar to macrophages. So, we call them as macrophage-like cells. Now, again, monocytes, as I told you, they circulate in the blood after leaving the bone marrow. They only survive for a day or so before they enter the tissue and finally mature into a macrophage. They are involved in phagocytosis and intracellular killing of microorganisms. Monocytes or macrophages are having antigen processing and presenting. That means the mechanism of antigen processing and presentation must occur in the macrophages. That is, antigen is taken up, it is broken down into small molecules, and this is called antigen processing, and then finally it is presented on the membrane. They have degradative enzymes in lysosomal granules, they chew the ingested proteins and they present it to the adaptive cells. Now, when monocytes enter the tissues, they become macrophages, they become enlarged and they increase the production of intracellular lysozymes, and in macrophage stage they can live for years in the tissue and they are highly motile within the tissue. Now, coming to specialized phagocytic or macrophagic cells, the dendritic cells. Now, from where these dendritic cells have got their name, as you can see in this slide, these dendritic cells have certain extensions just like the extension present in the nerve cells, the dendrites of the nerve cells, hence they are called the dendritic cells. They are found in most tissues, especially at interfaces between the external and the internal environment. For example, if it is present in skin, we call them as Langerhans cells. They can be present in the lining of GI tract where they encounter various invading pathogens. So, they are extremely important cells because these are the cells which will take up the pathogen, cleave it into small particles and then present it to the T cells. Once they presented to the T, whether to the T helper cell or the T cytotoxic cell, that is a CTL cell, it does not matter. The dendritic cell are very important to present it to, mostly to the T helper cells. For example, if it is a APC or it is an antigen presenting cell, dendritic cell, then it is almost exclusively presenting it to the T helper cell. They are extremely motile and continuously they check their surrounding with the help of these endocytic processes or dendrites. They are efficient at activation of T cells. The further T cell development is dependent on them to control responses to the antigens. Now, as I told you, there are various types. If it is present in the skin, this is called Langerhans. If it is present within the organs, it is called interstitial, interdigitating, or if it is just present circulating, we call them as circulating dendritic cells. Now, there is another species of dendritic cells which is called follicular dendritic cell, which is not like the normal dendritic cell, but instead what does follicular dendritic cells does? It checks the immune complexes present within the blood. The immune complexes comes and bind to the membrane of the follicular dendritic cell, and in this stage the immune complex can stay within the blood for a longer duration. Now, when the immune complex is staying within the body for a longer duration, the body will have more time to produce a effective immune response. So, it increases the it decreases the clearance of the immune complex by adhering to the immune complexes floating around in the blood. So, follicular dendritic cells are much different from the other types of interdigitating, interstitial or Langerhans type of dendritic cells. Now, coming to the natural killer cell, a very important lymphocytic cell. The natural killer cells are also called large granular lymphocytes or LGLs. They do not express the normal membrane molecules or receptors that are present on the T lymphocyte or the B lymphocytes. Now, the purpose of natural killer cells, now as you can see from the slide, the natural killer cells are much bigger. They have much more cytoplasm and they are granular. They are not agranular, they are rather granular in nature, and they kill the virus infected or the transformed cells. Now, natural killer cells have actually two types of receptors, the KIR receptor and the CAR receptor. The KIR is the killer inhibitory receptor, while the CAR are the killing activator receptor. Now, target recognition occurs by killer inhibitory receptor which is lacking on the infected or tumor targets. Now, what happens? The infected, virally infected cell, it is your own body cell. It will have MHC molecule, MHC1, MHC2, that is a major histocompatibility complex antigens would be present on these because they are your own body cells. Now, what will happen if it is a normal body cell? It will have the normal amount of MHC protein on its MHC receptors or proteins on its membrane. When an NK cell comes in contact with a normal body cell, the KIR receptors will get activated, that is the killer inhibitory receptor, which will tell NK cell not to kill the cell because this is a normal body cell. However, if your body cell is getting infected by a virus is residing inside, or it has undergone some transformation, that is, it has become tumorigenic. Now, once it has changed because its nature, because of presence of a virus or it has undergone transformation, the amount of MHC on the membrane of your own cell will change. Once this amount of MHC changes, the CAR, that is the killing activator receptor of the NK cell, will get activated and will tell NK cell to now kill this tumor, the transformed or the virally infected cell. So, the body ensures by means of NK cells that all those cells which have gone hayway, which are not right, which are not normal, which have gone abnormal, whether due to virus or due to transformation, they are removed through the NK cells. So, they kill targets by releasing perforin. Now, they are, as I told you, they are granular cells, they have granules. The granules have perforins and granzyme. Whenever a transformed cell or a virally infected, your own body cell is coming in contact with the NK cell, the perforant punch holes into this cell, the target cell. After punching holes, the granzyme goes inside this cell, the wrong cell, and kill this cell. So, by means of perforance and granzymes, the NK cell finally kill the wrong cell, the transformed cell, the virally infected your own body cell. There is another process where NK cells are involved, which is called ADCC, or antibody dependent cell mediated cytotoxicity. This process takes place because NK cells have CD16 receptors on their membrane. Now, what happens if an antigen is present? Most of the times an IgG is produced against this. This is an antibody G is produced against it, which goes and binds to the antigenic determinant on the antigen. Now, when this antigen antibody complex is formed, now the CD16 is a receptor which binds to the Fc region of the IgG. This complex can come and bind to the CD16 receptor present on the NK cell. So, antibody is kind of forming a sandwich where on one side is an antigen, the other side is the NK cell. Now, once this complex is formed like this is called antibody dependent cell mediated cytotoxicity. The NK cell will now kill the antigen. There is another species of cell called NK1 T cell that has some of the characteristics of both T cells and NK cells that has also been recognized in nature. Now, coming to the next, that is the basophils and the mast cells. The basophils are found in the blood in low numbers. They are involved in type 1 hypersensitivity responses. They have high affinity to Fc receptor for IgE. As you remember, IgE is the immunoglobulin which is responsible for the hypersensitivity or allergic responses. Now, cross-linking of IgE to its receptor on the basophil leads to the release of certain pharmacologically active mediators like histamine, prostaglandins, leukotrienes, which lead to, in a mild form, lead to an allergic reaction, or in a in a very bad form, it is called an anaphylactic shock. Similar goes for the mast cells as basophil, but mast cells are exclusively present in the tissues. The stimulation of mast cells also occur by anaphylatoxins, that is the complement protein C3 or C5a, or by cross-linking of IgE on its surface receptor. Once IgE cross-links on the surface receptor of either mast cell or basophils, these anaphylotoxic material is released, histamine etc., and will cause a lot of allergic reaction or sometimes the anaphylactic shock in the organism. Now, the eosinophils, they, as we know, they are the acid staining. They are intensely stained with eosin. They have a characteristic bilobe nucleus. They mediate toxic reaction to large parasites and are involved in asthma. Eosinophils are motile, sometimes phagocytic, and are particularly active in helminthic reaction. They release a toxic which is called major basic protein, which helps in removal of the parasite from the body. Now, we said there are another types of cells which are involved in the adaptive immune response. These are the lymphocytes, the B cells, which are called the plasma cells or the antibody producing cells, and the T cells, which can be called as cytotoxic T lymphocytes or the T helper cells. Now, T helper cells are further Th1, Th2, Th17, T regulatory cells, T suppressor cells, etc. Now, lymphocytes constitute about 20 to 40% of the body's white blood cells and 99% of cells in the lymph. They circulate in the blood and the lymph and are capable of migrating into the tissue spaces and lymphoid organs. They have the most important immunological attributes, that is, these are the attributes of the adaptive immunity, that is diversity, specificity, memory, self and non-self recognition. Now, the markers present on the T cells which distinguish whether it is a T cell or T lymphocyte or a B lymphocyte, as you can see, the marker which is present on the B cell are CD19, CD20, CD40 along with the immunoglobulin molecule which are present on the surface. While in case of CTLs, that is T cytotoxic cell, you have CD3, CD8 and of course the TCR, which is the T cell receptor. In T helper cell, you have the T cell receptor along with CD3 and CD4 molecules. Now, what is important in adaptive immunity? It is the specificity. which resides with the antigen receptors which are present on the T and the B cells. Now, the TCR, that is the T cell receptor or the B cell receptor, both are specific for only one antigenic determinant or epitope. Therefore, T cell is having a receptor which is monovalent, which binds to only one determinant present on the antigen. However, the BCR that is B cell receptor is divalent, because you have two arms of the antibody which can recognize the same epitope, right. So, BCR is bivalent while TCR is monovalent. Now, coming to B lymphocytes, they develop from stem cells in the bone marrow. They produce antibodies with specificity for the antigen. Now, plasma cells can be formed by antigen binding to the surface immunoglobulin, which activate the B cells into a factories of antibody producing plasma cells. On activation, a B cell can have class switching and can produce a different class of antibody with the same antigen specificity. They are the most efficient and presenting cells, antigen presenting cells of the body also. Now, on the other side, the T lymphocytes, they undergo development in the thymus. That is why they are called T lymphocytes. T stands for thymus. They are involved in the cell mediated immunity. Now, important part is that the T helper cell influences the P lymphocyte to produce antibody. So, hence they are called T helper cells, because they render help to the B lymphocyte to produce a specific antibody to a specific antigenic epitope. The mature T cells display either CD4 if it is T helper cell, or it display CD8 if it is a T cytotoxic cell. The cells with the CD4 marker, as I told you, are called T helper cell, while if the cells with CD8 marker are called T cytotoxic cell. Besides, it also have a TCR, which is called T cell receptor molecule that recognizes antigen. TCR is a transmembrane heterodimer which is composed of two polypeptide chains. Now, as you can see, what happens in T helper cell, they express CD4 molecule which helps in the cell mediated immunity. Now, when the antigen comes, it is taken up by an antigen presenting cell, it is cleaved, that is processing takes place, it is then presenting as you can see, the one is marked as antigen associated with the MHC present on the antigen presenting cell. This is an antigenic peptide which is getting bound to the MHC molecule, and now it is being recognized by the T helper cell through the CD4 molecule. Now, there are various phenotypic population of T helper cell, as I told you, T helper 1, T helper 2, etc. Coming to T cytotoxic cells, they are cytotoxic against tumor cells and the host cell infected with the intracellular pathogen. So, you must understand, in the T helper cell we have antigen presenting cell, right. Antigen presenting cell are specialized cell which are picking up antigen and showing it to the T helper cell. In this T cytotoxic cell, it is our own body cell which has gone wrong. It could be a tumor cell, it could be a cell infected with an intracellular pathogen. It takes up the pathogen and after processing and presentation along with class 1 MHC shows it to the CD8 molecule of the T cytotoxic cell, which now has become CTL, that is cytotoxic T lymphocyte. So, here it expresses CD8 molecule receptor which helps in the recognition of the antigen molecule. We also have T suppressor and T regulatory cell. Not much is known about them. T suppressor cells are supposed to suppress T and B cell responses, while T regulatory cell are again a subpopulation of T helper cell which regulate this T cell responses. Not much is known about them. Coming to the antigen presenting cell, they are found in the skin, lymph nodes, spleen and thymus. They present antigen to antigen sensitive lymphocytes. They include macrophages, B lymphocyte and dendritic cells. They have two properties, that is, they express class 2 MHC molecule on their membrane and they are able to deliver a co-stimulatory signal that is very important for the T helper cell activation. Now, coming lastly to the two lone cells, that is the RBC and platelets. The erythrocyte also have some role in the immune system. They bind to some small immune complexes and help them to remove them from the blood circulation. On the other hand, the platelets release certain mediators when they get activated. These mediators activate complement proteins, leading to attraction of leukocytes at the site. Now, with this I would like to end my lecture. Thank you very much.
On that note, I would like to thank ma'am for this very enriching discussion, and I would like to thank you dear friends for watching our show. I hope you have come to understand what immune cells and the system is all about. On that note, thank you dear friends for watching our show. Stay tuned and keep watching. Thank you.